All right, great. I think we can get started today. Thank you all for joining us today at the Dana Farber Cancer Institute Connect to Science Seminar Series. A couple of quick reminders before we get started. I just want to encourage everyone to visit the website listed at the bottom of Larry's slide here on the shown on the screen in order to get updates on the Connect to Science series, including getting a list of upcoming speakers. Also, during the course of the presentation, if you have a question, please just post it to the Q&A section at the bottom of the screen instead of putting it into the chat box. So I'm thrilled today to be hosting our speaker, Larry Boyes, who is the R. Randall Rollins Chair in Oncology in the Department of Hematology and Medical Oncology at Emory University School of Medicine. Larry earned his PhD in pharmacology at Virginia Medical College and pursued postdoctoral training at the Howard Hughes Medical Institute at University of Michigan, as well as University of Chicago. It's actually at the University of Chicago, working with Craig Thompson, that Larry discovered the gene that encodes the anti-apoptotic protein BCL-XL, which is being widely pursued today as a target for cancer therapy with great promise. Larry initially held a faculty position at University of Miami Miller School of Medicine, which is where I was fortunate enough to meet him and be inspired to pursue a career in apoptotic cell death regulation. Larry's pursued his research in the fields of cancer therapeutics and heme malignancies for many years, has been incredibly successful, and has helped bring many new therapies to patients diagnosed with multiple myeloma. He's also received multiple awards for his research, as well as mentorship, including the Mentoring Award from Emory University School of Medicine, which I'm sure he's quite proud of. Today, we'll hear about his latest research in multiple myeloma and his continued pursuit of improved therapies. Larry, thank you so much for joining us today. We're looking forward to hearing your talk.
Chris, thank you for that kind introduction, and of course for the invitation to speak today. It's a real honor to be here today. And so I am going to talk a little bit about our work in myeloma and a lot of the pursuit that we've had in thinking about how to treat this disease, as well as potential mechanisms of resistance to therapy. And I will actually at the end talk a little bit about some functional precision approaches that actually we've talked about. So, but before I get started, these are just my disclosures. And I want to, while I was working in Craig's lab, he did teach me that you got to show your most important slide up front so you don't forget it, and that is the thanks. And I just want to mention the people in the lab, and I'll try to mention them all throughout the talk, but the work has really been, that I'm going to focus on today, was really led by three members of the lab, Shannon Matulos, Vikas Gupta, and Ben Barwick. And I'm pleased to announce that both Vikas and Ben have now moved on, are now assistant professors, and they both have their own K awards, and that's great. And but there were many others involved, and I will mention them as long as we go. And working on myeloma, I'm really privileged to work at Emory and the Winship Cancer Institute with this wonderful team. And I will feature a little of the work with Mala Shanmugam and Jonathan Kaufman as well. And we haven't worked in a vacuum, and we've had wonderful collaborators across the country over the years. And I'm going to particularly focus on some work today with Konstantin Mitsiadis, Dana Farber, Jonathan Keats at TGen, as well as, and I should mention Kelvin Lee, who we've really been working on a lot of this together, probably for almost 20 years now. And Kelvin was also a postdoc in Craig's lab, but we started that later at Miami. He's now at Indiana University. Our funding sources, of course, and most importantly, our patients and their families, as we use a lot of patient samples through this, and it's advanced our research, and hopefully we've been able to do that to maybe help them as well. And so, in thinking about myeloma, I'm always drawn back to this poem. This was written by Yash Mehta, who was a high school student, a summer high school student, 17 years old when he worked in the lab. And he started a seminar, much to our surprise, with when he finished up his 6 weeks, with this poem. And I'm not going to read the poem now. You can probably read it faster than I can. But he hit on all the real main points here, which is that what is multiple myeloma? It is a malignancy of the plasma cell. And the symptoms are related to effects in the bone marrow, where you have anemia due to crowding of cells, because you have this plasma cell malfunction, as shown here in the growth of these malignant plasma cells. those actually activate osteoclast and inhibit osteoblasts, and you get these lytic bone lesions as shown down here in these pictures, as well as with all the immunoglobulin secretion, you get renal dysfunction, and we can measure the immunoglobulin secretion in the blood as it's a monoclonal protein, as a clonal disease, and you see this as known as this M spike in the serum peptide protein electrophoresis. And Yash was quite optimistic that it was research that was going to be this cancer's main foe, and probably all cancer's main foe. And he had, I think, reason for optimism. There was a lot of new things happening in myeloma at the time. It was a little over 10 years ago. We didn't have antibodies or CAR T cells or bispecifics, and now we have a lot of those that are have been approved for the treatment of this disease. So I think this to me is still the best way for me to think about what myeloma looks like clinically. Genomically, it looks more like this, and this is just a surplus plot taken from a study I'm going to talk about, the MMRF COMPASS study, looking at these are copy number variations or changes in about 800 newly diagnosed myeloma patients, with the red being gains and the blue being loss. And you can see there's a very unusual pattern in myeloma where about 50% of the patients have what we call hyperdiploid disease, because they have trisomies of kind of odd numbered chromosomes. So 3, 5, 7, 9, 11, 15, 19, and 21. And again, that happens in about half of the patients. So that'll become relevant in a little bit. We have other recurrent changes. So on chromosome 1, we have a loss of the 1p and gain of 1q. And I guess important in thinking about lots of cancers is what's happening on 17p, where we do have loss of 17p, where p53 is as well. But copy number isn't the only changes that are occurring in myeloma. And so in these same patients, we can look at the frequency of single nucleotide variations. And the RAS-RAF pathway is probably the most common of these types of mutations, with about almost 50% of patients having either activating KRAS, NRAS, or BRAF mutations. However, we do have these p53 mutations and some other NF-kappa B non-canonical activating mutations as well. But the real drivers in myeloma are these translocations. And so those are shown here as the IGH heavy chain translocations, particularly to cyclin D1, to a gene, the 414 translocation, which results in the activation of both this gene, which is now called NSD2, as well as FGFR3 in about 3/4 of the patients who have a 414 translocation, and MAF and MAF-B. And these are really, these occur in about 50% of the patients. So you usually either have a IGH translocation or hyperdiploid disease, but there are lots of other translocations as well. And that's going to be a little bit of the focus of at least the first part of this talk. And so when does this all happen? It all is part of a germinal center reaction, where we have a naive B cell here showing being activated by something that looks like a COVID particle, although this figure was actually made in 2019, so it was not COVID at the time. And upon this activation, B cells go through that in a T-dependent fashion, go through this germinal center reaction, which results in affinity maturation of the immunoglobulin through somatic hypermutation, as well as class switching from IgM to a different effector function for IgA or IgE, et cetera. And the fate of these more efficient B cells is they can either recycle back into memory and actually re-enter this potentially, or they become long-lived plasma cells in the bone marrow, where they stop growing and they just live there and can be quite long-lived in the case of some vaccines, which have shown us. And so what typically happens in myeloma is one can envision that there's a lot of genomic changes happening with mutations and rearrangements due to the germinal center reaction, and sometimes these will there'll be dysfunction in this and resulting in these IGH translocations. However, these cells, even though it happens in the germinal center, still move to the bone marrow. And so they become a plasma cell that can secrete antibody. And again, it'll be a monoclonal because these cells now have acquired the ability to start growing and through and also other changes. So some of these other things that I mentioned, like the AMP1Q can hear deletion and the different types of mutations that occur, and that we see that that these tend to be later events, is that we see that in these IGH or hyperdiploid or early events found in premalignant cases or asymptomatic, are known as smoldering myeloma, while we tend to acquire these other mutations, some earlier than others, through the diagnosis of myeloma and the relapse of this. And so how is this, how do we think about myeloma? And this is something that's really inspired some of the work that I'm going to show today. Was that, originally we just thought of myeloma as being this malignancy of plasma cells. And then as I showed you, we end up finding that there are these really sort of two classes of myeloma, the hyperdiploid myeloma and the non-hyperdiploid that have these IGH translocations. So we split myeloma into a few more classes because there's several of the IGH translocations. And then with gene expression profiling, I know it's hard to see here, but myeloma is now split into 7 to 10 different categories. And then you add on the mutations that I just talked about, you have quite a heterogeneous disease here with only any of those translocations occurring in about 15 to 20% of patients, and even the RAS mutations each are about 20% for KRAS and NRAS. And where our dilemma was, and this is going back about 10 years, was that about the same time that we were learning this, we also saw this was happening in treatment of myeloma. So this was data from the Mayo Clinic from Shaji Kumar. And what he showed was from 1971 to 2006, and really from 1971 to about 1971 to 94, there were, despite any kind of changes in myeloma therapy, nothing was happening. And then all of a sudden in 94, you start to see the line starts to move. And by 2001 to 2006, it's really moved. And now through work, actually some of it done here by my clinical colleagues, it's way up here in some cases. And so the question was that how is this happening without any new molecularly targeted personalized therapies, knowing that all these changes that we were finding in myeloma was splitting myeloma into lots of different groups. And so none of the success that we saw in these therapies during this time were actually being informed by what we're learning in this genomics. These tended to help with prognosis, but they did not tend to we weren't targeting those changes. And so that led us to start thinking about, well, what are the potential functional dependencies of myeloma, and something that would be common to all the myeloma. So kind of taking us back to this plasma cell. And so this was just a kind of a silly exercise we did using some data that was from where all of what was then Affymetrix array that was publicly available, grouping the different cell types to find out were there genes that were specifically associated with a cell. And so this just happens to be the plasma cell data. And so you can see that there were 488 genes that were found expressed in 95% of all the publicly available data at that time on Affymetrix arrays. And if you convert that into functions, there were 214 functions, of which 272, of which 214 were actually found in everything. So these were really kind of housekeeping type of genes and functions. And so that left 58 functions, gene ontology functions that were associated with being a plasma cell. And what were those? And so you got an immune cell that was highly differentiated, associated with cell structures that are related to protein folding and assembly and RNA stability and survival, again, long-term, long-lived cell, and other signaling types of responses. And so this really kind of changed the way we started thinking about myeloma, and that maybe this was a disease of two biologies, where you had all these changes associated with a cancer biology. However, there were still functional dependencies associated with being a plasma cell. And so this is sort of was from a review we wrote almost 10 years ago now, the Kelvin Lee and I with Nizar Bayless and Jonathan Kaufman and Sagar Lonial. And this is from a cell line, MM1S, and this was just structural changes we found in some whole genome sequencing at the time. And this is the same cell line. This is an EM taken by a former student, Esther Obeng, of the same. And what you can see is you can see the nucleus right here. And what you can see, and even at this late stage of being a cell line, there's all this endoplasmic reticulum that's kind of running all through here. And this still makes actually an Ig light chain. So it's maintained even as a cell line, many of the characteristics of a normal plasma cell. And so based on this, we postulated that probably those dependencies are what the drugs were targeting that were resulting in better survival of these patients at that time. So we started adding drugs like these IMiD drugs and proteasome inhibitors, and we're just starting to test drugs after that that were targeting actually the cell surface, and these are all plasma cell surface markers that we now target with antibodies or bispecifics or CAR T cells. The IMiDs we now know from work of Ben Ebert and Emil Kalin, they were working really through the changing of the targeting of this colon ligase complex so that they targeted these transcription factors that are important in B cell lineage, Ikaros and Aleos. And we had been working on the idea that the sensitivity of proteasomes was really related to immunoglobulin secretion as well. And those, I think, have all stood the test of time as being important here. We've now added drugs that we can include as based on the genetic targets, but these, I would say, have not been as a big a hit as some of the ones that target this plasma cell biology, and will most likely be used in concert with those moving forward. And so we've been left with the question of how do you target that yin of myeloma, that plasma cell biology? And this is really what we've been working on for probably about 15 years now. And so we think of these cells as protein folding machines, so anything that alters protein catabolism. I mentioned Esther's work there. Transcription factors that maintain the plasma cell state. And I'll talk a little bit more about Ikaros and Aleos in a little bit. But we've had a lot of wonderful collaborations here with Nizar Bayless and Paolo Neri and Leif Bergsegel and Marta Casey in this area. My background is in BCL2, so obviously long-lived cells, we think about BCL2 inhibitors, and I'll talk a little more about that. And then finally, and through a really long-standing collaboration with Kelvin Lee, again, we were both postdocs together, we've been thinking about about the bone marrow microenvironment and interactions with other cells, such as dendritic cells, or as well as some stromal cells, although I'm not really going to take any time to talk about that. But it's a most recent collaboration with Konstantin Mitsiadis and several labs that he's put together. And we've been working on this project for 5 years that has really, I think, solidified some of these ideas. And so this is some of the work from a paper that I think is getting close to being across the finish line. But Konstantin was generous enough to allow me to show some of the work. And this is work looking at trying to define preferential dependencies in a disease. And so using the DepMap data, comparing the myeloma cell lines to all the non-myeloma cell lines and looking for dependencies which are defined by SIRA scores of anything that was blue would be considered a dependency in myeloma. And there were 116 of these genes that statistically were preferentially dependent. So it doesn't mean they were exclusively dependent in myeloma, but they were more dependent in myeloma than if you compare it to all the other diseases. And what did they end up what are these dependencies? And so again, kind of like what I just showed you about plasma cells, we end up having transcriptional regulators, many of which are in fact related to lineage. So the number one, these are shown in the order of the dependency. And the number one here is IRF4, PAL2AF2, and PRDM1 are all three B cell and plasma cell lineage transcription factors. We then get into the antibody secretion, so unfolded protein response, Golgi, transport from the ER, and mRNA regulation, just like we saw in the plasma cell. I'll skip down some of the metabolic changes, again, associated with antibody production, but it could also be associated with growth and signaling molecules. So a lot of this looked awfully similar. And indeed, there are a lot of things that here in this list, the dependencies that were related to being a plasma cell. And I think this is further exemplified by this circus plot that Ricardo and Yosuke worked on in Konstantin's lab. And I call it the least interesting circos plot ever, and that it takes those 116 dependencies and maps them out, and then asks the question of how do these dependencies look if you rank them against changes associated with disease, so mutations, deletions, amplifications, whether they're come up in TCGA data sets and different myeloma sets for progression-free survival in different trials. And so in this circus plot, if the gene ranked, it was either going to be, if it was in the top 50 or 100, it's going to fall in this yellow sort of orange, or if it's a highest percentage, it's going to be red. And what you can see is there's very little yellow or orange or any or red beyond that first circle that just shows the serous dependency. So most of these are not altered in myeloma. And so it's more of a dependency related on function. And so why do we see that in myeloma? I think some of it's related to the differentiation statement. So this is doing the same analysis, but using other diseases. And what you can see is that myeloma has the most preferential dependencies. You have to correct a little bit for the number of cell lines in the depth map for each of these. And so, because that would have an effect on the statistics. But if you just compare myeloma to other diseases that have very similar number of cell lines, it's way higher. And indeed, the solid tumors, actually, you see very little dependency. I think the median here for the solid tumors is about 5 compared to some of the leukemias and lymphomas that are shown here. So I think that the reason myeloma is still even higher than these is probably related to the fact that it's not just cell of origin, but it's that differentiated cell of origin as shown here that's even maintained when you are a cell line. I always thought it was curious that melanoma actually came up as the highest hit of the solid tumors here, which maintains some of its differentiation. And so what I'm going to do now is just tell a couple stories about how we've used this to and thoughts about this to think about responses to drug therapy or new therapies. And the first story I'm going to talk about is how, in fact, changes in the cancer biology actually had a significant effect on targeting the plasma cell biology. And in fact, and that's going to really focus on these drugs, these IMiDs like lenalidomide and thalidomide, and how specific alterations affected this, and really by affecting a very specific aspect of the plasma cell biology. And the second story I'm going to tell is a BCL2 story, but it's going to be one about how an altered differentiation during plasma cell, in the plasma cells, the myeloma plasma cells, actually changes the dependency of the cells onto BCL2. And I'm going to finish up with a little bit of some functional precision medicine that we've done based on that. And I would say that we weren't planning on doing functional precision medicine, and we kind of just found our way there. But if you want to really hear about some really exciting approaches in functional precision medicine, I really highly recommend that you check out the monthly seminar series from the Society of Functional Precision Medicine. It's online, and it's been a great series. So with that, we'll start with the first story about the IMiD resistance. And this really was made possible by a large study from the Multiple Myeloma Research Foundation called the COMPASS study. And this was a study the clinical part was led by my colleague Sagar Lonial here, and Jonathan Keats at TGen did all the genomic parts of this. And the idea, there was no TCGA project in myeloma. And so what they decided to do was kind of do their own. So they took what was originally supposed to be 1,000 untreated myeloma patients. It ended up being a little bit more than that. And using then getting comprehensive genome molecular profiling, so whole genome, exome, and RNA-seq on these patients at diagnosis, and then following them every 3 months clinically, as well as getting samples at relapses. And originally it's supposed to be followed over 8 years, but it actually ended up going a little bit longer than that as well. And we were really excited about the whole genome sequencing aspect of this, because this was an opportunity to have that level of whole genome sequencing really gave us a chance to look more deeply at those structural events associated in myeloma. And so this was really work led by Ben Barwick, who I mentioned in the beginning. And this was the inner circle of that first circles plot I showed you, showing the translocations that were found in at least 1% of the patients out of the 795 that we had data on for whole genome on this. And again, you can see the red actually represents the clonal events, and the black are the ones that are not clonal. And this is just spread out to show that if you look at who had hyperdiploid disease, you can see that these are not enriched where you have IGH translocations. As I mentioned, about half the patients have IGH translocations and half of them are hyperdiploid. There is a little bit of overlap there, but overall, 2/3 of the patients had some kind of translocation. They just weren't all IGH. And so you can see here, if we look at these IGL translocations, mostly to MYC is shown here, IGL translocations are actually found primarily where they're in the hyperdiploid population, not where there tend to be IGH translocations. It's true of the MYC translocations, where the majority of them consistent with work from earlier work from Leifberg, Sagel, and Mike Keel. On this. And so what do these translocations look like? This is actually looking at where the breakpoints were. And what one can see is in the heavy chain, you can see that they break very consistent for the most part with the breakpoints that you would have for class switch recombination, suggesting they're involved in that, but not all of them were. Sometimes some of them fell in between. We had some in the VDJ regions as well, which could suggest an earlier event, but while they're clonal, it also is possible that these are complex translocations that have picked up multiple regions there. One of the things that we did learn from this is that at these different breakpoints, it's very interesting that almost all of these were 14, 16 translocations at the A2 breakpoint, where we see very different with some of the other IGH translocations. This is a translocation of the MAF gene that occurs here. And curiously enough, in those patients, we actually see a lot more IgA2. And so it suggests that while the chromatin is clearly opening at this locus during class switching, whatever is driving the class switching to A2 is probably also inducing those changes somewhere near that MAF locus. And that's why you tend to get those types of translocations during that time. So what did this all mean clinically? And we always thought about these MAF translocations as being a little bit worse, but within this data set, we actually didn't see any changes in a real significant difference between progression-free and overall survival. And there could be several reasons for that, but we decided that was a good reason to move on to other translocations. So we looked at the other side of the story. The other side of the story was MYC. And so MYC is the most commonly translocated gene, even though not necessarily just to IGH. And again, these tend to be later. And all these translocations result in an increase in expression of MYC compared to not having a translocation, or a little bit compared to copy number. But all these are fairly equal. So whether you're translocated to the IGH, the IGL, or the IGK. But curiously, it was only the IGL translocation that resulted in a decrease in progression-free and overall survival. And so that forced us to really rethink and look more carefully at the IGL translocations. And sure enough, they seem to have a worse prognosis. About 10% of patients have one. And so what was different about these patients? Well, it wasn't age, and it wasn't their staging at diagnosis, and it wasn't the demographics or how they were treated, or the mutations that were found. And we found a slight difference in gene expression, but one difference we really found was in structural variation. And I kind of alluded to this already, which was that 70% of the IGL translocations co-occurred with the hyperdiploid disease. So this is just that circus plot kind of spread out into a map and showing that's where all of these IGL translocations occurred. And so why is that important? Well, generally speaking, we think of of hyperdiploid as being, one of the better prognosis for myeloma, more of a standard risk disease. And indeed, if you take the hyperdiploid patients and you take out the IGL translocations, you can see that they do significantly better than the rest of the population. But if you look at the ones that have an IGL translocation, they're doing worse. And so we don't test for IGL at this point. And so that suggests that in these 10% of patients, they're being misdiagnosed as being a standard risk when they actually have a high risk marker that nobody's really looking at. And so we wanted to look more about what that marker was. And what we found was that we could separate patients. About 2/3 of the patients received IMiDs. Almost everyone received bortezomib or a proteasome inhibitor. So we could look at IMiD versus no IMiD. And clearly patients who get an IMiD did better than those that didn't here, unless those were patients that had an IGL translocation. And so if you had it, there was no effect there. And so it suggested that the patients who had these IGL translocations were resistant to IMiD therapy. We don't see this when we look at the IGH translocations. So this was not something generalizable. It was really had to do with this particular translocation. And we wanted to ask mechanistically why that was. And so teaming up with Nizar Bayless and Paola Neri, what Ben did is analyze some ChIP-seq data for Icaros, again, the target of IMiDs and that are degraded during that. And this is looking at the IGL locus and the super enhancer that is found there. And one can see that if we look at the IgH, K, or L and these 3 different cell lines, that there's significantly higher levels of Icaros bound to the IGL. So how does that influence the response? And so what Paola did was she actually treated cells with lenalidomide prior to doing the ChIP-seq. And this is looking at 2 loci. So one of them is this DUS22 enhancer, and this is what drives IRF4, which was the highest preferential dependency in myeloma. And here's the IGL translocation. And what one can see is, first of all, is that these are on different scales. So this is 2.5 times higher. When you add lenalidomide, you do drive the levels of both of them down by about half of what they were, but half here is still as high as this started on the other locus. And the consequence of that is when one looks at the binding of BRD4 or P300 at those enhancer sites, is that while those are decrease in accordance with the changes in Icaros at the DUS22 site, there's actually no change on the P300 site. So these are still transcriptionally active. And so if an oncogene like MYC is being driven there, it's not being shut off. And so to summarize to this point, we looked at these nearly 800 patients and identified a new marker of that were associated with poor prognosis as IGL, about 10% of the patients. And it was in a group of patients primarily that we would have thought had a good disease. And it appears that this is related to their inability to respond to IMiDs and due to the binding of Icaros to that site. And so where do we go from here? And so one of the nice things about COMPASS was there were about 114 African-American patients where we had whole genome sequencing as well. And so we actually compared the structural variants in African-Americans, where it's twice as more common a disease than in Caucasian patients. And one can see that while the IGH translocations are fairly similar in their how common they are, we do see some differences in these more rare translocations, and that are their non-IGL translocations. And in addition, there's some differences in response. So this is just looking at the IGL translocations in white versus African-American patients and the IGL-MYC, and you can see that they do significantly worse. So we've recently been funded to further examine some of these non-IGH translocations in African-American patients. And so with that, I'll move to the second story. And so this one, again, is related to BCL2. And we've always thought about how to treat patients with BCL2 inhibitors, but we also use these in many ways as great chemical biology tools to study dependency in the lab on why cells can express 3 different anti-apoptotics, in this case, many myeloma cells, but maybe only be dependent on one of them. But for clinical purpose right now, there have been tests with MCL1 inhibitors, and based on the plasma cell biology, we would have thought MCL1 would have been a great target for myeloma. We've been limited somewhat to the use of venetoclax here, but it's been a very exciting journey. And this is the original phase 1 study of venetoclax in myeloma, and it was designed for all comers in this study, and there was a 21% overall response rate. But what one can see is that based on preclinical data, primarily from Martine Amiot, there was some thought that patients who had this T1114 translocation, so the cyclin D1 translocation, might actually benefit more than other patients. And so centers like ours, and Jonathan Kaufman really leading the charge here, really just focused on putting patients who had 1114 translocations on. So while not initially designed to be a precision medicine trial, it morphed into one, as one can see that almost half the patients actually had the 1114 translocation. And there was where all the responses were, with 40% response in that group versus only a 6% response in the others. And based on this, we also in the lab started thinking about what could we combine venetoclax with, and Shannon Matulis did some very nice studies showing that dexamethasone made a really good partner for reasons it's all published, so I won't go into now. And Jonathan and I approached AbbVie about opening an expansion cohort for this original trial and just doing 1114 patients with venetoclax and dexamethasone. We got it up to 60% overall response, and then a phase 2 was a little lower, but these were more heavily pretreated patient population. But so we had a precision medicine marker 1114, but it's still about a coin flip. And so we were wondering, was there a way to improve that? And so Vikas Gupta really took charge here, and he looked at 31 different myeloma cell lines and did dose response curves. And what he found was pretty much exactly what we found in the patients, which was that 1114, about half of them were sensitive and half were resistant. And in non-1114, only about 10% of the ones that he tested were sensitive. And so why was that? And when we looked at BCL2 family mRNA expression of the BCL2, BCLX, and MCL1, one can see that, yes, BCL2 was significantly higher in the sensitive cells, shown in green, and while BCLX was borderline lower and MCL1 was a little bit lower in the sensitive. And you can really expand these differences by doing ratios, and one can see that you can get some really high ratios. And there's been some association of these BCL2, BCLX ratios with sensitivity. However, in half of the sensitive patients, the ratios are no different than the resistant ones. So we didn't think that this was a particularly useful marker, at least at the mRNA level. We wanted to learn more about what the differences were beyond BCL2. And so what Vikas did is he analyzed RNA-seq data from the 31 cell lines, as well as from a cohort of patient samples that were by ex vivo testing, either sensitive, shown in green, or not, or resistant, shown in orange. And it was comforting that when he looked at the genes that were differentially upregulated in the sensitive cell lines and created a gene set from this, that gene set was highly enriched in the patient samples, genes that were also upregulated. And when he looked at this by comparing actually the cell lines to the primary samples, anything shown in black here was significantly changed, either up or down in this case, but only those in red were significantly up or down in both. So there was a lot of trending here, but only a few that were significant. And surprisingly, none of the ones that are significantly shown in red here start with the words like BCL or MCL or anything like that. And indeed, when we looked at the patient samples, we really didn't see these types of differences, except in the ratios when you get to these sort of extreme levels where you can make those kinds of calls. So, and the ratio works if it's a really big difference, but overall it was not predictive at the RNA level. So what were these genes and what were the changes? And somewhat surprisingly, what we found was that in both the cell lines and the patient samples, we saw upregulation of genes that were more associated with B cells. And in fact, a group of myelomas that have some B cell markers is defined by Frank Zahn and John Shaughnessy at Arkansas many years ago. And indeed, these genes, MC4A1 is CD20, which we tend to think of as a more B cell marker, not a myeloma one. So there was seemed to be an altered differentiation state that was being associated with venetoclax sensitivity. And so the first thing we asked is, well, are they really plasma cells? And if we look at the expression of plasma cell lineage markers, they clearly had expression of these transcription factors. When we looked at some of these other genes like CD20 or CD79 or other B cell associated genes, we can see that there was upregulation, although it wasn't consistent. You can see there were 3 cells lines that I had here, 3 that were here, and maybe 3 here, but they weren't all the same line. So this was not a normal differentiation state. There seemed to be just something altered, and some of these markers were upregulated or not. And this is easier to see in a Western blot where we're looking at the expression of some of these B cell markers in just cell lines that are 11-14. And one can see that if you look at the sensitive lines, they all express at least one. There's very low levels of STAT5A here, but only one strongly expresses all 3 of these markers. And in the resistant, only one expressed 1 marker. Sorry about that. Vikas extended this to surface markers, and he picked several plasma cell markers, one that can be found 86 on both plasma cells and B cells, and several B cell markers. And it was quite comforting that these markers actually in an unsupervised clustering by flow cytometry, they segregated this way, and they also segregated the sensitive and the resistant. So what Vikas did is he used that to generate an algorithm using 5 of the most I would say differentially expressed ones of here. He applied the algorithm back to the cell lines, which is a bit circular, but one can see that it was pretty good at predicting the sensitivity of this, and then applied it to patient samples. And again, it was actually very good with an 82% sensitivity of predicting based on an ex vivo testing of the cells. I will say that in 8 of these samples came from patients that did receive venetoclax, and both the ex vivo test and the flow prediction predicted 7 out of 8 of them. And so what was different about these? And we thought about it as being a differentiation issue, that we go back and think about the chromatin. We performed ATAC-seq on many of these cell lines. And I'm not going to show you all the data on the ATAC-seq, just that after doing some footprinting based on that and looking at sites that were in these differentially chromatin accessible regions in sensitive versus resistant, there were these enriched for these BATF or BZIP type sites. And so with Konstantin Mitsiadis, again, we did CRISPR activation on BATF and showed that we could sensitize a resistant line. And this happened in the absence of any changes in the protein expression of any of these BCL2 family members shown here. So it's changing the functional dependency without necessarily changing the expression. We're interested in how that happens. I'm quite fortunate that the lab right next to mine belongs to Mala Shamagan, and she's been looking at this in the context of metabolic dependencies, where she took many of the same cell lines that Vikas showed to be Ven resistant or Ven sensitive, and showed that the Ven sensitive ones had lower complex 1 activity, and that if you then treated with drugs that inhibited complex 1, you could actually sensitize both patient samples as well as cell lines to this. And the same thing was true of complex 2, where there was much lower complex 2 activity in the Ven sensitive. So this appears to be an electron transport issue that's quite different. And in patient samples, again, there was this correlation between SQR activity and venetoclax sensitivity. So again, potentially a mechanistic explanation to why these somewhat altered differentiation cells have this effect. And so I'm going to move on for the last, try to move on in the last minute or a couple minutes or so, and go back to this graph of the venetoclax phase 1 study, and tell how this kind of got us into doing some functional precision medicine with this. And that was that we were actually using these drugs as chemical tools and testing lots of samples. And it just turned out that 2 of these samples right here up at the top, the patient who had the stringent CR and the other CR patient, we had tested it, and they were very sensitive compared to other ones with venetoclax. And so we started looking at this and found that indeed, when we looked at a series of patient samples tested ex vivo for a 24-hour and 11.5 killing assay that it looked an awful lot like what happened in the patients. And again, 1114, about half of them, we use 100 nanomolar IC50 cutoff in this assay. About half of them were sensitive and half of them weren't, and about only maybe less than 10% of the non-1114s. So this is what that group of patients actually looked like if we spread them out. And there were some natural gaps in this where there was almost a 2 to 3-fold difference right here. And so we called these the sensitive ones and painted them green, and painted these intermediate ones yellow, although we don't really think about them as that much anymore, as I'll explain in a second. And the red ones were the really resistant, and anything that had an asterisk had an 1114. And just turned out that 16 of these patients or so were treated with venetoclax. So we had a pre-treatment sample here, and one can see that within the sensitive ones, all our responses were within the sensitive, and indeed about 90% of them, 9 out of 10 of them had a response with one with stable disease. And then when you got into the yellow and the red, there were no responses at all. And so we wanted to look more carefully at this. And so this was just some examples of how this was happening and how it was really this ex vivo sensitivity was dynamic. And so this was a patient that we got a sample and it tested in this intermediate group over here. And then 2 years later, we got another sample, and it was quite sensitive. And this patient actually ended up going on venetoclax, and right before, it was actually the sample to go on that clinical trial. And you can see that they had this wonderful complete remission or complete response to venetoclax. Unfortunately, all the stories didn't have happy endings. This is one where we got an initial sample. It was quite sensitive. A year later, the patient came back. We got another sample after the patient went on venetoclax, but it was really resistant, and the patient actually ended up progressing very rapidly in this. We had many samples where we got a pre and a post, and again, in almost every case that we saw this, we saw that the post sample was more resistant than the pre sample, or we had a post sample. It was a relapse sample. They were resistant. And so and one of the ones I want to point your attention to is this MM91. We thought, well, we did a further testing on this, and we thought, okay, well, if it's becoming really resistant to a BCL2 inhibitor, it might be sensitive to an MCL1 inhibitor. And it actually was the most resistant sample we'd ever seen to an MCL1 inhibitor to date still. And so we wanted to learn more about what that was. And so this was a patient actually of these Arvalis, and they did single cell CNV copy number on this. And we figured, not surprisingly, based on what we've done, there was no changes in MCL1 whatsoever. And so it wasn't an amplification of that, but there was a mutation in BCL2, and this mutation conferred resistance to venetoclax when put into a cell. And it did so because the mutation, when you had it, could still bind to proapoptotic proteins like BIM, but the drug could no longer displace it. And this is very similar to some of the mutations that have been observed in refractory CLL. And so we've updated our data again on the responses for patients. And these are only patients that receive venetoclax or venetoclax and dexamethasone. And we still see 90% of our sensitive cells respond. Our resistant ones, we now started to see some responses, and I'll get to that in a second. However, with added time, we can now see that this actually offers a test predicts a better progression-free survival with a specificity that's consistent with a biomarker using a 24-hour test. So what happened in these other patients? And so we also had done most of them had received venetoclax and dexamethasone, and so we actually had tested ex vivo that, and you can see in 2 of them, there was a significant sensitization to the venetoclax with the dexamethasone. We didn't have data on this last one. And then I think in the last slide, what I'll just show you is we went back to think about other potential biomarkers to compare this to. And so this is looking at RNA expression in most of those patients that we had RNA-seq done. And we really didn't see anything except, again, one patient with an outlier, maybe 2, ratio here at the RNA level. But there was no correlation with progression-free survival within these patients, and no advantage in survival. And probably most telling was that when we compared to patients where we could do IHC and look at BCL2 protein, there was no correlation with message and protein. So how did the protein do? Actually, pretty much as well as the ex vivo test did. And if we have both, it really does quite well. And so from this, we would conclude that the venetoclax sensitivity is associated with these B cell markers in myeloma. And this does provide a unique opportunity to use phenotypic profiling as a marker. That these B cell markers are heterogeneous. And so it suggests that this is not a normal immature plasma cell state or a progenitor state, but this is some kind of altered or inappropriate differentiation. And that for test purposes, we can't use just the one antibody because the CD20, for example, did not actually was not predictive on its own. And it looks like we're gonna have to do a panel similar to what's been done in lymphoma. And the ex vivo testing does extremely well, just as well as the IHC did here. And it's nice because you can get it done in 24 hours and it's probably less subjective than some of the IHC. However, it's obviously limited to academic centers. And so we've got to learn how to reduce this to a simpler practice. And I think that's really one of the challenges in functional precision medicine right now. But there's, again, some beautiful work going on. So with that, again, I thank you for the opportunity, and I'd be happy to take any questions.
That was great. Thank you so much, Larry. So just to remind everyone in the audience, if you have questions, please just type them into the Q&A section at the bottom. I see that there's some questions coming in. But I'll take this first opportunity just to ask a quick one, because I know that there's been a lot of work done on trying to understand how metabolic changes may sensitize cells to BH3 mimetics, including venetoclax, including work that you contributed to as you showed in the presentation. I'm wondering, like, what is the potential clinical strategy for manipulating metabolism of these cancer cells and sensitizing to venetoclax? Are there good potential strategies, and what's being pursued for multiple myeloma?
Yeah, so I think there have been several studies where they're using combination type of approaches ex vivo and looking for things. I think one of the challenges with that is some of the drugs that end up, so dexamethasone has come up in a few of them. It came up in ours, and it came up in one that Keith Stewart did. I know others, HDAC inhibitors have come up quite a few times. There's been some challenges with that. As that they're pretty toxic, and so that I don't know if that's going to move forward. But again, the question is, how do we? I like the Latai approach of doing some, and I should say in the Sorosiak approach too, of doing some of the BH3 profiling, particularly the dynamic BH3 profiling, as a way to try to define some of those, or what drugs can make cells more BCL2 dependent. I think that's going to would be an outstanding way to do it.
Speaking of Tony, he typed in a question here. He goes, it says, "Great survey, Larry. What are the barriers keeping you from deploying the functional precision medicine into everyday care of myeloma patients?"
Yeah, again, I think I think this would be the approach to do it. I think we could do it in an academic setting right now. We just have to get the test certified, so I think we could do that. The question is that going to be something practical moving forward? And so one of the areas that we've been working on, I'm very fortunate in that at Emory we have a shared biomedical engineering department with Georgia Tech. And there's a lot of people who've done a lot of different types of point of care testing in different aspects. And so I've been in early discussions with them and trying to think about how do we reduce this to a simple test that could be done at any clinic and not just in an academic, research setting. I do think for some of the functional precision medicine, it's going to be a trial like this that's really would push it forward. I love the work that has come out of Austria and Helsinki and such, but those are those are really going to be limited, I think, to academic settings right now, where they test multiple drugs and such. Beautiful work.
Makes sense. Then we have a question from Zintis Indy. Is the B-cell marker resistance mechanism you described found mostly in primary venetoclax resistance or in acquired resistance as well?
Yeah, that's a great question. So again, so there's the B, if you have the B-cell markers, you tend to be sensitive. And so the question is, do you lose those, I guess, really in resistance? And the answer, so Nizar Vallis's lab showed some data on this actually at the ASH meeting last year, and it looked like the answer was no, that the resistance mechanisms are probably unrelated to that.
Okay, great. Next question is from Gareth Morgan. Do you see consistent differences in gene expression or drug sensitivity when IgH expression is lost, either before diagnosis or at relapse?
So IgH expression. So we're talking. I guess we're specifically thinking about the antibody here, not a translocation. There does seem to be some associated with what we call non-secretory disease. So a lot of patients will go through a stage where they actually lose their heavy chain, and so they'll just make a light chain and secrete a light chain, or they'll even lose that, and so they become less and less plasma cell-like. And I do think you do see differences in some of the therapies that end up affecting that target that plasma cell biology. It's hard to prove in the lab because you could just silence the heavy chain or the light chain, but that doesn't really change the whole biology of the cell. It just takes away the one marker. And so I think that's not a real a good way to test that. But I think the answer, I believe, is yes, that you do see differences there. It's just a different phenotype of the cell.
Neat. Another question is, you went through the different lineage-based dependencies in multiple myeloma. Are there other things on the horizon that are coming up as targets for multiple myeloma? Again, coming back to that plasma cell biology and the vulnerabilities associated with those cells.
Yeah, I think this is a great question, Chris, because what I mentioned earlier, we haven't targeted we still go back to the cancer biology of it, and so those translocations and many of cyclin D1 doesn't come up as a preferential dependency because it's a dependency in too many other cancers. Cyclin D2 does, and it's induced by the MAF translocations, and MAF itself was up as a dependency, and FGFR3 was on there as a dependency as part of the 414 translocation. We just really haven't done a great job of targeting those, and one is left to wonder if some of the new ways that we can target drugs where we don't necessarily target genes where we don't have to necessarily inhibit them, so through degraders and such like that, if that's not going to offer an opportunity to really go back at the sort of the core mechanisms related to the pathogenesis of the disease.
Yeah, that makes sense. And another question that is particularly interesting to me, what do you think is the future of MCL1 inhibition in multiple myeloma? What do you think about the not only toxicities, but then also, the on-target effects of MCL1 inhibitors on myeloma cells? Do you think that there's a future there? If so, what is it?
Yeah, it's interesting to me as well. We always knew there was a lot of promise because of the lineage, and that we knew, from her, early work, and that these were in normal plasma cells, that this was going to be, that was likely to target. It's also on chromosome 1q, which is is amplified in myeloma, so it's upregulated. It's not only there, the dependencies there, it's upregulated, and there was a lot of preclinical work, but from work of Joe Offerman and such, we knew that there was also that chance of toxicity and cardiotoxicity based on his developmental work that he'd published several years ago. And, unfortunately at this point, most of the inhibitors, we've run into a little bit of those types of toxicities. So I don't know if that means that we just need better MCL1 inhibitors that are better, have even a better affinity than the ones we have, so that we can use less of it to try to get around that toxicity issue. But it's, it is teasing to be that close and not quite have advanced yet with the MCL1 inhibitor.
I think it's going to be interesting next few years with MCL1 inhibition and myeloma. Hopefully we can solve that problem one way or another.
Yeah, there's always the possibility of coming up with ways of directing it to the cells as well. And I know that there's been some work with that with some other inhibitors. So maybe that's going to be the way around it.
Absolutely. Wonderful. Well, thank you so much, Larry. We really enjoyed your talk. This was a real pleasure seeing you again. Thank you so much, everybody, for tuning in to this Connect to Science seminar series, and we look forward to seeing everybody again next week.
Thanks,
Take care,
Chris.
Larry. Thank you so much.
Take care.